Alrighty, welcome back, everybody. Welcome back. So here we are starting chapter 4. Chapter 4 is going to be covering the biology of the cell. So you look at this little funny cartoon and you'll see what we're talking about. All right, so you can see the learning objective that we have for every single chapter that you're familiar with by now. So I'm not going to go over them, just take a look at those so you know what's going to be expected of you on the exam. So let's review the hierarchy in biology. And remember, we talked about the atom and the molecule and the macromolecules. So we've covered chapters 2 and 3, discussing the atom, then molecule, then macromolecules. Now chapter 4 is going to cover the organelle that are the organelles that are found in the cells. So before we can talk about the cells, we've got to talk about cell theory. So remember, we reviewed this cell theory. We covered this already. Cell theory, 1, all living organisms are composed of 1 or more cells. 2, cells are the smallest unit of life. And 3, new cells come only from pre-existing cells by cell division. So remember that and keep that in mind. Very, very important. So the cell, let's talk about the component of the cell. So this is a really nice a micrograph of a normal light microscope of your epithelial cheek cells. And you can see here that we have the cytoplasm, and you can see the nucleus, which is an organelle. You can see a cell membrane. And since this is an animal cell, there is no cell wall. But you can also see these amazing tiny little structures in there. And these are the bacteria, normal flora bacteria that you find in your cheek cells. So we're going to be going over the parts of a cell, starting with or talking about prokaryotic cells and then moving into eukaryotic cells. So remember, on the tree of life or the web of life, we have the prokaryotic species, which include the bacteria and the archaea. And then we have the eukaryotic organisms. The bacteria and the archaea species fall under the category of prokaryotic cells. So you can see that there are multiple structures that are found in the prokaryotic cell. We have the nucleoid, which is the region where DNA is found. You have ribosomes, you have a cell membrane, you have a cell wall, you also have some pili and flagella. Some bacteria have flagella on there, some bacteria have pili on there for reproduction. We also have the cytoplasm, where you find the ribosomes and where you have the site of metabolism. So you can see a colorized transmission electron micrograph, TEM, of E. coli, Escherichia coli, which is a bug that is found in our gut. It's part of our normal flora of our gut. Okay. Looking back on the web of life, you can see eukaryotes. The difference between prokaryotes literally mean before nucleus. Eukaryotes means true nucleus. We're also going to see organelles, and remember that there's compartmentalization. Okay, there are lots of eukaryotic species in this class, but we're going to talk about 2 major ones, the animals and the plants, in this chapter. All right, you can see the animal cell, much more complex than the prokaryotic cell. You have a membrane-bound organelle known as the nucleus. You have membrane-bound rough endoplasmic reticulum, membrane-bound smooth endoplasmic reticulum, membrane-bound Golgi complex, membrane-bound mitochondria. So all of these structures you're going to see are responsible for different functions in the cell. Plant cells, the difference, they are circled in red. You can see there's a central vacuole, which helps to control water. There's a chloroplast, which is the site of photosynthesis. And you have the cell wall, which provides additional structural support for the cell. Okay, and we're going to talk about these structures individually. Okay, animal cells, plant cells. You've talked about this. I've helped my son in this when he was in 4th grade, and he covered it again in 6th grade. Then he went again over it again in 8th grade, and then he had high school biology last year, and I covered that with him. So again, you've seen this over and over, so there's not much we can do with it, but I do just want you to be aware of the parts of a cell and the difference between the animal cell and the plant cell, and the difference between prokaryotic and eukaryotic organisms. Okay, so the cellular components that we're going to talk about, the cytosol in the cytoskeleton, the endomembrane system that we're going to be talking about, a lot of structures that are composed of membranes. Semi-autonomous energy processing structures, which we're going to talk about later, and then external structures, cell wall, the cilia and flagella for movement, the extracellular matrix, and we're going to talk about finally cell connections. Okay, so again, another little cartoon here. All the organelles supposedly communicating with one another. There is communication, but obviously not in the cartoon style that you're seeing here. Okay. All right. So please just take a look at those previous slides and review before you move on to the details. Okay. So let's start. Cytosol and the cytoskeleton. So the cytosol is basically the colloid, the solution, the suspension, which is mostly water, that is found in the cell. So this is contained within the cell. The cytoskeleton has the protein fibers throughout the cell, and those are moved by using motor proteins, for example, kinesin, dynein, actin, and myosin, which we'll talk about a great deal in physiology, the actin, myosin, how that works to contract the muscle fiber, and how you get movement. The microtubules, intermediate fibers, filaments, excuse me, and the actin filaments. Basically, looking at them, they have different functions. Basically, these help to maintain the structural integrity of the cell. So cytoskeleton, basically, you can think of it like the steel beams that are found in a building when you're constructing a building. You have the cytoskeleton, the skeleton, the infrastructure of the building, okay? You can see that these structures will help basically anchor the different organelles. They work together to obviously maintain the shape of the cell. So remember, the shape relates to, everybody should know this by now, it's like beating a dead horse. Yes, function. So again, these structures help to maintain the shape of the cell so that it can perform its specific function. Okay, so you can see all these images in the table talking about them and comparing. Just take a look at those. You'll cover these more in upper level cell biology courses and again. Okay, the endomembrane system is a fascinating system. Again, this came about through evolution. And you have to remember that the prokaryotic cells do not have the endomembrane system. So when the eukaryotic cells evolved from the prokaryotic cells, they had new, basically, again, just like you saw in the hierarchy, there was an element that was not present there before. So you didn't have membrane-bound organelles in the prokaryotic cells. Now we have a nuclear envelope, so a nucleus. You have endoplasmic reticulum, which has a smooth and divided into rough, we're going to talk about. You have a Golgi apparatus or Golgi complex. You have plasma membrane. You have a lysosome. You have the vacuoles. Again, all these evolved from our prokaryotic ancestors. Good? All right. So let's talk about the nucleus. The nucleus, okay, it contains most of an organism's DNA. We say most because we're going to talk about another organelle that also has DNA in it. okay? For the, again, the eukaryotic organism, the chromosomes, the DNA is actually linear, okay? And it's wrapped in a nuclear envelope. And the nuclear envelope is made up of a double phospholipid membrane. You have nuclear pores, which we're going to talk about in great detail in Unit 4. So that means something is going to be able to go in and out of the nucleus, okay? Very important for the DNA. The function, basically the nucleus, because you have one, it's a location, the site of DNA replication, and that's basically where you take one DNA and you make two. It's also the site of transcription, which is RNA synthesis, where you take a DNA molecule and you produce mRNA, messenger RNA. In the nucleolus, that's a structure found within the nucleus, you have the process of basically producing ribosomes. So ribosomes that are found in the cell, in the eukaryotic cells, are basically synthesized in the nucleolus of the eukaryotic cells. And that nucleolus also contains our RNA, ribosomal RNA. So ribosomal RNA aids in producing or synthesizing ribosomes. Again, we're going to talk about that later on. The rough endoplasmic reticulum, basically, it's the site of protein sorting. It is called rough because it is studded with ribosomes, okay? So basically, you have the synthesis of secreted and membrane-bound proteins, okay? You also have glycosylation occurring, okay? So basically, it's where you're going to metabolize sugar. The ribosomes are going to be very, very important. We're going to talk about those in great detail also later on. They're basically the site of translation. Translation is protein synthesis. You build the ribosomes, or they're assembled in the nucleolus, but then you can find them after they're synthesized, either freely floating, they'd be freely floating in the cytoplasm. Okay, so cytoplasm is the cytosol, the liquid, plus the organelles. So then the free ribosomes are going to be involved in the creation of proteins. And those proteins are going to function within the cell. For example, enzymes are going to be used in cellular respiration, which we're going to talk about in chapter 7. The bound ribosomes, which are attached to the rough endoplasmic reticulum, they are going to create proteins also, or synthesize is the better word. I like the word synthesizing proteins, that will either be leave the cell, so they're going to leave the cell and go somewhere else, or they're going to be part of the membrane of the cell. So for example, they may possibly go to the mitochondria, and they'll go to the inner membrane of mitochondria, where they will be involved in the process of generating ATP. So again, you can see we're magnifying right here. You can see a electron micrograph of the rough endoplasmic reticulum. Obviously, it's stained. The blue part is the endoplasmic reticulum, and then these brown little spots are the ribosomes that are bound on there. Pretty amazing, fascinating. Yes, all this stuff is real. You just got to be able to zoom in and look at it, okay? A smooth endoplasmic reticulum, very important function. Basically, you have the synthesis of lipids there. You also have different metabolic functions there in a smooth ER, detoxification. So the smooth ER helps to break down different drugs that we ingest. For example, alcohol is a big one, any toxins that come in. So you can imagine, you've heard which organ is involved in detox or breaking down alcohol. Yep, you guessed it, the liver. So you can probably imagine liver cells have a lot of smooth ER in them, because it's going to be working overtime to get rid of that alcohol that you consume. Okay. We also have the storage of calcium ions in the muscle cells. So in the muscle cells, it becomes a sarcoplasmic reticulum, and you release calcium in order to have the contraction occur, which will generate the movement that we see. For example, me speaking right now to you, moving my hands, walking around, so on and so forth, whatever it may be. So the smooth ER stand for endoplasmic reticulum. All right, next we have the Golgi or Golgi complex or Golgi apparatus. Basically, the function is to take in vesicles that are carrying, for example, proteins. They have two faces. They have a cis face, which is the receiving end, and then you have a trans face, which is the transport end. So basically, I think of the Golgi complex like Amazon, okay, the Amazon center, distribution center. You order something, it comes in, it's at the Amazon, they package it, get it ready, and then that's going to be on the cis face. And then on the trans face, you have the truck standing by to deliver the packages to your front door. Okay, so basically, the Golgi complex is going to take part in a process known as post-translational modification, basically modifying the proteins after they have been synthesized. All right. Next, again, we can see that the endomembrane system is involved in the production of a protein, starting from a beginning and going to the end. And again, this is a really nice figure because it goes over the processes of what happens to a protein when it is produced. And again, you can just see what a protein goes through before it can become functional and it becomes ready to do its job. So proteins. You're going to be hearing me talk about proteins the entire semester for all of 181 because they are just so important. So I'm going to come back to this later on, but I did want to put this in here to show you, just give you a sample of what's to come. Okay, lysosomes and peroxisomes, they're created by rough endoplasmic reticulum and they're processed by the Golgi. Basically, lysosomes, I like to think of them as the garbage trucks of the cell. They go around, they'll pick up anything that's not supposed to be there, anything that's worn out, anything that's outlived its usefulness, okay? You dump it into the lysosome. The lysosome has what we call hydrolytic acids, and basically they will break down those substances that should not be in the cell, that the cell no longer wishes to keep. Okay, pretty amazing. They're going around and they're cleaning the cells constantly. Okay, so very, very, very important. Peroxisomes are involved in anabolic and catabolic reactions we're going to be talking about. And then it produces basically catalase, which helps to break down hydrogen peroxide. So we'll talk about that later on. All right, so if you look at the endomembrane review, you start off with the nucleus with a nucleolus, then you go to the rough ER, then you go to a smooth ER, then you have a transport vesicle, which then will take you to the Golgi complex. And then from the Golgi complex, it's transported to whatever location it needs. Or if it's outlived its usefulness, you have the amazing lysosome. Pick it up and get rid of it. So basically, it's like cleaning your curb up if you dump out an old couch, okay? Garbage truck, bulk pickup will come around, okay, once a month, right? And, you know, the day it's gonna be, all right, so basically, comes and picks up your couch that you don't use anymore, and it's gone. It's the same thing with lysosome, but it doesn't wait once a week. It is functioning all the time. It's a 24-hour day, 7-day a week service in the cell, okay? So much more efficient than our trash pickup, right? All right. Vacuoles. Vacuoles basically are membrane-bound sacs of whatever. They could be anything. They could be gas, they could be water, they could be digestive, they could be pigment, they can be poisons, okay, depending on what you are, okay? So those are vacuoles. You can see there are numerous types of vacuoles. You can see central vacuole, you can see contractile vacuoles in either a plant cell, for example, or in or other organisms that have contractile vacuoles found within them, okay? The semi-autonomous organelles, these two are absolutely fascinating, and there are courses just dedicated to this. The mitochondria and the chloroplast, all right? So we're gonna talk about these. The mitochondria, we're gonna have a long discussion about the mitochondria in chapter 6 and chapter 7, and then we're gonna have a long discussion about the chloroplast in chapter 8, but I do wanna give you an introduction to it. The mitochondria basically is involved in the final steps of cellular respiration. It is a double membrane organelle, okay? The inner membrane folds into an area known as the cristae, and basically that will increase the surface area. So again, because you are involved in the process of cellular respiration, we're going to talk about what that means and the fact that you can produce this enormous amount of energy in this organelle, okay? So, the unique thing about mitochondria, they also contain DNA. This is what's known as mitochondrial DNA. If anybody's ever watched Forensic Files or CSI or Law and Order SVU or NCIS, I've seen multiple episodes from all those TV shows. CSI Miami, all have had episodes where the criminal has been caught because of mitochondrial DNA, okay? The chloroplast, again, these are very unique organelles. They are found only in plants. And basically, they are involved in the process of photosynthesis, okay? And we're going to talk in great detail about this when we get to chapter 8. But the thylakoids have the pigment known as chlorophyll in them, and that's how you're going to be able to take the energy from the sun, which comes in the form of light, and then you can convert it into ATP, which is a form of chemical energy, and we're going to talk about all of that, don't worry, in our second unit, okay? Again, it is a double membrane bound organelle. This also, believe it or not, has DNA. So it has DNA, its own DNA. So how can these two structures have their own DNA? Well, we have what's known as an endosymbiosis theory. And the theory is this: the mitochondria and the chloroplasts originated from bacteria that took up residence within a primordial eukaryotic cell. That means that originally the pre-eukaryotic cell, okay, and these mitochondrial and chloroplast ancestors, okay, were once prokaryotic organisms, and they merged together and became part of the eukaryotic cell. How can we identify that, and why do we have that theory? How will we be able to come up with that theory? By observation, right? It's the purest, simplest thing that we do as scientists, observing. The mitochondria in chloroplast are semi-autonomous. They contain their own genes. So that means they have DNA. And they also divide by binary fission. What structure did we talk about? Divide by binary fission? Oh, right. Dr. E mentioned it. Bacteria. Okay. And the mitochondria are very structurally similar to some of the bacterial organisms that we still find on our planet today. Absolutely fascinating if you think about it. Okay. So you can see the animal cell, you can see the plant cell. Here's the endosymbiosis theory. Okay, you have a purple bacterium, you have a cyanobacterium. It merged a little bit. Okay, again, pre-eukaryotic. These are prokaryotic. These are pre-eukaryotic. So in between here, they're eukaryotic. So we're talking about billions of years ago, okay? About from here to here, about the time, maybe 1.5 billion years ago, this occurred, okay? This basically, this endosymbiotic relationship between the pre-eukaryotic cells and the prokaryotic cells that would eventually become the mitochondria and the chloroplast. Just to give you a little bit to think about, okay? It's really, really cool. All right, so you can see again, just going through in the ancestral and the way that they basically became a part of our present day eukaryotic cell. So if we're looking at this cell, okay, what is this? It's going to be a plant cell, okay, because it has a chloroplast, and you can hear it's probably an animal cell, chloroplast, but they're the mitochondria. All right, external structures. Okay, we talked about the internal structures, and that's what everybody learns about in grade school, and that's what gets all the fanfare. But guess what? There are really amazing external structures too that are really important in helping to maintain the structural integrity of the cell. Maintaining the structure is so important in biology. If you don't know that by now, in the 2 weeks that I've been talking, then there's something wrong, because you know I've mentioned this multiple times. Structure relates to function. We need to maintain the structure of the cell in order for it to function properly. So the external structure we're going to be talking about, the cell wall, the motility structures, so those are structures that aid with movement, and then the extracellular matrix, so those are going to be found in animal cells. All right, cell walls. Where do we find cell walls? Well, the plants have them. They're comprised of cellulose, which we talked about, the polysaccharide. Prokaryotes, they have glycopeptides. It's the glycocalyx. Basically, it's found in the cell wall of bacterial organisms. Others have silica, chitin, for example. The crustaceans, lobsters, crabs, for example, have other different types of cell walls. So a lot of variety. That's the beauty of biology. Variability, variety, right? So the cell wall in plants, again, you can see it had a primary and a secondary. Why would you need a cell wall in plants? Well, plants have to be able to regulate water. And if there's an overabundance of water, okay, it needs to be able to handle it. So you have the central vacuoles, but you also have the cell wall, which will help to maintain the structural integrity of the plant cell, allowing the plant, the tree, the shrub, the bush, whatever it is, to stay alive if there's excess water or in drought conditions when there's not enough water. It's going to help maintain its structure in either of those situations. We'll talk about that in 182. Motility structures, for example, the cilia, So cilia that you find basically on paramecium, but then there's that helps the paramecium to move as a single-celled organism, or the cilia that are found inside of our respiratory passages. For example, in our nose, the nasal passageway to our throat, the pharynx, down into our trachea. Again, it helps with movement, but it also helps to trap particles. The flagellum, here's a magnification of the amazing sperm cell. So this is how we came about. That's how we're alive right now, the sperm cell, right from our dads, okay? So you could see that the nuclear material is contained in here, and everybody knows the stories of how the sperm swim to the egg to fertilize it. So it swims using the tail, quote unquote, and the tail is actually the flagella. Finally, the extracellular matrix. This is found in animal cells. So for example, all of our cells have extracellular matrix, and basically, just like the cell wall in plant cells helps to maintain the structural integrity of the cell and maintain its shape, the extracellular matrix in animal cells helps to, again, strengthen and support, bind cell signaling, help to maintain the structural integrity and basically the shape of the cell. The components of the extracellular matrix, you have adhesive proteins, you have glycoproteins, polysaccharides that work. Again, all of this will be discussed in upper level courses, okay? But I just want you to know that the extracellular matrix is comparable to the cell wall in plants. So plant cells do not have extracellular matrix. Animal cells do, because we need the extracellular matrix to help maintain the shape of our cells. All right, so glycocalyx, again, we'll talk more about these later on. Cell junctions, I am going to skip this. I'm not going to put this on your exam because I talk about this in the first week of Bio 201, which is anatomy and physiology. So for those of you that will be taking A&P here at ASU West, we'll be talking about it in great detail. So I'm just going to skip these slides right now and just move on and just tell you about the review. I told you it's going to be a short chapter because you've all seen this material before. There's nothing new here. Okay, so review the prokaryotic cells, review the eukaryotic cells. Okay, so all this are just review slides for you. Go over the animal cell and all the structures that we talked about and the organelles. Really, really important. Plant cells, go over them, right? They're starred structures that you will not see in animal cells. That's why they're starred here. The centrovacuole, the chloroplast, the cell wall, the plasmodesma, okay? And then you have the comparison of bacterial cells, animal cells, and plant cells. A really nice table to help you organize yourself when you're prepping for the exam. Okay, but again, don't just memorize this table. Remember, be able to interpret information from this table. Study guide terms, again, remember, put them on note cards. It's going to help, but remember that you don't just memorize. You're going to be like, wait a minute. All right, oh, RNA, ribosome RNA. Oh, okay, well, what does that have to do with the I'm going to remember bound ribosome. Okay, oh, wait. Doesn't rRNA make the ribosome, and rRNA makes the ribosome in the nucleolus? So see how you have to be able to move around. So don't just memorize. You have to be able to tell the story of how everything occurs. All right? Concepts, there you go. Really easy, one slide. Just go through those. Do not worry about 14 and 15 here. Do not worry about 14 and 15 under the study guides concept slide that I'm on right now. So 1 through 13, excuse me, is going to be sufficient in preparation for this exam. So just go through the organelle review again. Nice and laid out for you. All right. Ladies and gentlemen, that is chapter 4. I promised you it's going to be a short chapter, very straightforward, very easy. You've seen this numerous times before. So hope that it helped clarify some stuff that maybe you've seen in high school biology. And I hope that you can get together with your classmates and study. And then if you do have any questions about this lecture, please make sure to email me or to ask questions in the Zoom meeting. All right, until next time, this is Dr. E signing off on this video.